Hi everyone, welcome to my A-level biology lecture series. In this lesson, we will explore glycolysis. At the end of this lesson, you will be able to outline glycolysis as phosphorylation of glucose and the subsequent splitting of glucose 1,6-bisphosphate into two triose-phosphate molecules, which are then further oxidized to pyruvate with the production of ATP and reduced NAD. Let's get started. In glycolysis, the main process and main idea or the main purpose of glycolysis is to break down a molecule of 6-carbon glucose into 2-molecule 3-carbon pyruvate. From the name itself, glycolysis, glyco means sugar, lysis means split. So therefore, the entire reactions here focus on the splitting of the glucose, which is 6-carbon molecule, into 2 molecules of 3-carbon pyruvate. So the process takes place inside cytoplasm, which does not require oxygen, and we say that it is independent of oxygen concentration, regardless of whether oxygen absent or present. It will not affect the occurrence of glycolysis. Glycolysis consists of 2 distinct phases. First, energy investment phase, where we spend ATP to kickstart the glycolysis. At the end of the reactions, we're going to harvest 4 ATP molecules during the energy payoff phase. So therefore, it's quite similar to our business. For example, we start out a business by buying ingredients, we're buying some instrument and so on. We spend money first. Similar goes to glycolysis, where we start to spend ATP first, and towards the end, we harvest ATP. So for a glucose molecule, we spend 2 ATP during energy investment phase, and at the end of the process, we harvest 4 ATP molecules. Therefore, there is a net gains of 2 ATP molecule per glucose molecule. Glycolysis involves oxidation and reduction reaction. Let us look at this oxidation and reduction process. In general, when we talk about oxidation process takes place when a molecule or a substance, loses hydrogen atom or gain oxygen atom or lose electrons. On the other hand, when reduction process takes place on a molecule, that particular molecule gain hydrogen atom or oxygen atom is lost from the molecule, or this particular molecule gains the electrons. But during glycolysis, we will focus on hydrogen atom only. Let us look at this diagram. First of all, we have a substance or a molecule A with hydrogen atom. So hydrogen atom now is transferred to chemical B. So in this case, chemical A loses hydrogen atom. So therefore, chemical A is oxidized. On the other hand, because chemical B gains hydrogen atom, so chemical B is reduced. We are going to look at this concept later in glycolysis, as well as link reaction, Krebs cycle, and also oxidative phosphorylation, where oxidations and reduction take place during aerobic respiration. Let's look at this energy investment phase. During energy investment phase, we start with glucose molecule. Glucose molecule is generally non-reactive, quite stable, low energy stage. So therefore, glucose molecule will not spontaneously split, from 6-carbon glucose molecule to form 3-carbon triose phosphate. We need to supply energy. So one of the way to do so, we need to make glucose become reactive. One of the way to do so, we need ATP. So at first, ATP phosphorylate glucose molecule to form glucose 6-phosphate. So step 1 is termed as phosphorylation, and this is very important to make sure that we can increase the reactivity of glucose by phosphorylation process. ATP donate phosphate group to glucose so that glucose now accept one phosphate group to form glucose 6-phosphate. However, glucose 6-phosphate is still relatively non-reactive. It's not, the energy level is not high enough to split it into two molecules of triose phosphate. Therefore, we have a further phosphorylation where another molecule ATP phosphorylate glucose 6-phosphate to form fructose 1,6-bisphosphate. Okay? So step 2 again is termed as phosphorylation, where this glucose 6-phosphate is further phosphorylated by another molecule ATP to form fructose 1,6-bisphosphate. So now fructose 1,6-bisphosphate has sufficient energy. Therefore, fructose 1,6-bisphosphate is reactive enough to split into two molecules of triose phosphate. From 6 carbon, fructose 1,6-bisphosphate, now we manage to get triose phosphate during energy investment phase. So step 3 is termed as lysis, where fructose 1,6-bisphosphate is then split by the enzyme to form two molecules of 3 carbon triose phosphate. That marked the end of the energy investment phase, where two molecule ATP are used to phosphorylate glucose molecule to form triose phosphate. So after energy investment phase, followed by energy payoff phase. So at first, we have this triose phosphate, okay? So triose phosphate will be oxidized. In this case, triose phosphate donate hydrogen atom and NAD accept hydrogen atom. So triose phosphate is oxidized because triose phosphate loses hydrogen atom. How about NAD? NAD accept hydrogen atom. So therefore, NAD is reduced to form reduced NAD. So in step 4 is termed as dehydrogenation. From the term here, D means remove hydrogen atom from triose phosphate. So each triose phosphate is oxidized by giving out hydrogen atom, and NAD accept this hydrogen atom to form reduced NAD in step number 4. So after that, step number 5 and step number 6 are termed as substrate-linked phosphorylation, where phosphate group from the intermediate compound directly transferred, okay, to the ADP to form ATP. So we have gone through this process before, okay, where we call these processes as substrate-linked phosphorylation. A total of 4 ATP molecules are produced per glucose molecule when during energy payoff phase. So the end product in this reaction is pyruvate. Okay, so during a series of reaction during glycolysis, there are some chemical or some compound are produced. Let us look at what are the overall products of glycolysis per glucose molecule, or we say that when one glucose molecule is oxidized to form pyruvate during glycolysis. So there is a net gains of 2 ATP molecules. As we know, we spend 2 ATP molecules during energy investment phase, and 4 ATP molecules are produced during energy payoff phase. So a total of, or we say that a net gain of 2 ATP molecules per glucose molecule. And the end product, we know that pyruvate, and during the reactions, 2 molecules of reduced NAD are produced as well. Yep. Then, and the final product, pyruvates, are produced. Right? So 2 molecules of pyruvates are produced per glucose molecule. So this is extended learning where we're going to look at this glycolytic pathway step-by-step breakdown. So, yep. In fact, glycolysis is quite complicated, but for A-level syllabus, we just need to focus on glucose, phosphorylation of glucose from glucose 6-phosphate, and then another phosphorylation process takes place on glucose 6-phosphate to form fructose 1,6-bisphosphate, and with sufficient energy level, fructose 1,6-bisphosphate splits into 2 molecules of triose phosphate, and triose phosphate undergoes a series of reaction and oxidation to form pyruvate. So during the entire process, we spend 2 ATP molecules, and at the end, we gain back 4 ATP molecules. There is a net gains of 2 ATP molecule per glucose molecule. And as well as ATP molecule, 2 reduced NAD as well as pyruvates are produced during glycolysis. With that, we have come to the end of this lesson. I hope you have found this lesson helpful. If you have any question, feel free to leave them in the comments section. See you in the next lesson. Have a great day.